about the renal corpuscle. And the renal corpuscle is going to have that area, the glomerulus, and we're going to hit our loop, okay? The glomerulus is going to be the area that the blood is going to enter and the blood is going to exit. And it's going to be under some pretty high pressures in this area. So when we look at the renal corpuscle, this, don't remember if we got to this, but this is the example of the renal corpuscle. If you remember, when we begin to look at the blood flow in versus the blood flow out, having the structure that we look in this area, this sets up the situation for the blood flowing through these capillaries to be under high pressure. Because what that is doing, based upon this filtration membrane, okay, that we find around these capillaries, this filtration membrane, it's going to push materials out into this capsular area, and this fluid, which it's now termed, okay, because it's not blood, all right, once it gets pushed out, it's now fluid in the capsule, and it's going to make its way into the tubular area of the nephron. I think this is the point that we left off on Thursday. Now, as we begin to look at the tubular structure, the tubular structure, it's what is leading away from this glomerulus, the capsule that's surrounding it, okay? So note how they're trying to show my blood flow, the glomerulus, this being that capsular area that is now the fluid. So what we're going to find is we're going to see it leave that capsular area and begin to move through this tubular system. Now, what we're going to see is that we're going to travel through 4 regions of the tube. We're going to travel through what's called the proximal convoluted tubule, because that's what it looks like. We're going to hit this loop. We're going to have a descending limb, loop of Henle, ascending limb. This is going to lead to the distal convoluted tubule, and then to our collecting duct. So that fluid that was originally in Bowman's capsule has to travel the entire length of this tubular system. Now, the goal is that at each region of this tube, we're going to have some type of filtration occur. We're either going to pick something up, we're going to return something, okay, and make the way to this collecting duct, which makes its way into that, do you guys remember the renal pelvis area? All the calyces and so forth, okay? So that by the time we get to that point, urine is the composition it's supposed to be before it hits the ureter to leave the kidney. Note, they're trying to show on this collecting duct, they're trying to show this little part branching off, this part branching off, this part branching off. This one collecting duct will have several nephrons attached to it. So as this one is doing its job, this one would be doing its job, this one would be doing its job, this one would be doing its job, and it would all be coming into this collecting duct, therefore being a collecting duct. Does that make sense? Okay. This is what we get to see. Now remember, these are microscopic. They are very tiny. They are this functional unit of the kidney. And once again, I'm going to try to point out that we've got the two different types of nephrons. Ones that will be really close between the cortex and the medulla. Ones that are going to dip very deep into the medulla. Okay? And this, of course, being our collecting duct. If we take this loop and begin to kind of stretch it out so that we can have a view, note they're trying to show me my proximal convoluted tubule. They're trying to represent the flow of the fluid. Okay? So if we follow the flow of the fluid, and note that on this loop that exists, parts of it is thick, parts of it thin. So we have thick and thin segments. So we have our descending loop, our actual loop of Henle, our ascending limb. We meet into or drop into the distal convoluted tubule and then make its way into the collecting duct. Now that's going to be the flow of the fluid. At each portion, at the proximal, at the descending, at the ascending, at the distal, at the CD, something's going to be taking place for this filtration process that the kidneys do. The nephrons are also covered in those peritubular capillaries. The peritubular capillaries, in one set of the nephrons, they're here at the loop area. On the other set, they're found around the glomerulus area. Okay, just kind of keep that in mind, that not only do we have our glomerulus, the PCT, the loop, the DCT, the collecting duct, we're going to also find that we have peritubular capillaries. Don't forget those. So in looking at this tubular system, the tubule, the PCT, the proximal convoluted tubule. The proximal convoluted tubule comes from the capsule. That tube, or that portion of it, simple cuboidal epithelium. What do you remember about simple cuboidal epithelium? Simple tells me how many layers? One. Cuboidal tells me shape, okay? Epithelial, the tissue. What do you remember about a single layer of cells? What is it good for? For movement of substances. So the structure of the PCT is very well developed. The other thing that you're going to find on those cuboidal cells, a very prominent brush border. So those cube cells are going to have a lot of microvilli. They actually look like a hairbrush. That's why they're termed a brush border. This area has a great ability. It can do a lot of absorption. So at the point that that filtrate that fluid has now made it to the PCT. Why would this be good? Why would this structure, this makeup, be beneficial?
To transfer, like remove and add some substance.
Well, if we think about what has happened when we get to the PCT, in that glomerulus, where we had that capillary structure and the blood has come through and we've had it move in under that high pressure, there's a lot of stuff that has been moved into that space. Not everything that got pushed out into the fluid in that capsule needs to be lost. Some of the materials have got to be returned back to the body. Does that make sense?
Mm-hmm.
For the loop, the descending, ascending, the segments that are making it up, the thick segments, simple cuboidal epithelium. They're going to be in the thick segment, they're going to have the good ability for the transport of salts. The thin segment, simple squamous epithelium. What do you remember about simple squamous? Okay, it's the flat shape, looks like the fried eggs, okay? And if you remember, the thin segment was way down here on the bottom of that loop, okay? This one is going to be very permeable to water. The DCT, after we've gone through the PCT, around the loop, up to the DCT, cuboidal epithelium, but we don't have any microvilli on those. The collecting duct, if you remember, we're receiving from several nephrons, okay? The collecting duct has simple cuboidal epithelium. So that tells me some materials are going to get to move as fluid passes through or down that collecting duct. The collecting duct, it moves or converges towards the tip of that medullary pyramid. It forms what is termed the papillary duct. There's about 30 of them that end in pores at the tip of each papilla. And the urine is going to drain from these collections into that minor calyx. And then the minor calyx leads into a major calyx. So here's the flow of the fluid. The glomerular capillaries, from there to the PCT, proximal convoluted tubule, into the nephron loop, into the distal convoluted tubule, into the collecting duct, into the papillary duct, into the minor calyx, to the major calyx, to the renal pelvis. And do you remember how the ureter is attached to the renal pelvis, to the urinary bladder, to the urethra, and it exits the body? That's what it takes to form urine. Now, the formation of urine, well, that gets a tad bit interesting, to say the least, because the job is this filtration of the blood, the filtration of the waste from the body.